Hello students. So our chapter Human Health and Disease is going on and this is part 4. Today we'll study about acquired immunity and antibodies and types of antibodies and functions of antibodies in our body. So we'll be studying about all these things and the diagram of antibody. In the previous lecture, we have studied about immunity and types of immunity. Types of immunity are innate immunity and acquired immunity. We have studied innate immunity in detail. Today we'll study about acquired immunity. Acquired immunity basically is the immunity that an individual acquires after birth is called acquired or adaptive or specific immunity. It is specific and mediated by antibodies or lymphocytes or both, which make the antigen harmless. It not only relieves the victim of the infectious disease but also prevents its further attack in future. So the memory cells formed by B cells and T cells are the basis of acquired immunity. This acquired immunity consists of specialized B and T lymphocytes and antibodies. So we'll be studying about these in detail in next slides. T lymphocytes or T cells, they arise from bone marrow thymus. T cells form cell-mediated immune system. They defend against pathogens including protistans and fungus that enter the cells. They are formed by the division of lymphoblast of three types: killer, helper and suppressor cells. Lymphoblasts move to the site of infection. Killer cells react against transplants and cancer cells, and suppressor cells inhibit immune system. So these are what are T lymphocytes. B lymphocytes or B cells. These arise from bone marrow, bursa or fabricus in fowl. Gut-associated lymphoid tissue pairs patches. B cells form humoral or antibody-mediated immune system. They defend against viruses and bacteria that enter the blood and lymph. They are formed by division of plasma cells. Plasma cells don't move to the site of infection. Plasma cells don't react against transplants and cancer cells. Plasma cells have no inhibitory effect on immune system. We'll study about the what is antigen and what are its two properties. First of all, what is antigen? Anti means against and gen means birth, which is against birth. So definition of antigen is: antigens are the substances which, when introduced into the body, stimulate the production of antibodies. So it has two properties: antigenicity and immunogenicity. So the structure antigenic determinants are components of antigen. Each antigen carries many epitopes. So based upon the ability of antigens to carry out their function, antigens are of two types, and complete antigens and incomplete antigens. So antigens which are present on the body's own cells are called autoantigens, self-antigens. The antigens on the non-self cells are known as the foreign antigens or non-self antigens. So biochemically, antigens are proteins, large polysaccharides, lipids, fatty acids or nucleic acids. Sometimes entire pathogen acts as a. In this slide, we'll study about antibodies. Antibodies are immunoglobulins which are produced in the body in response to the antigen of foreign bodies. Thus, all antibodies are immunoglobulins, but all immunoglobulins are not antibodies. So, location and formation. The antibodies may be bound to a cell membrane or they may remain free. Antibodies are produced by B lymphocytes and plasma cells. In fact, B lymphocytes get transformed into plasma cells. The mature plasma cell produces antibodies at an extremely rapid rate. So types of antibodies are basically IgA, immunoglobulin A, IgD, and IgE, IgG, and IgM. So we'll study about all these in this slide. So first of all, I am telling you about IgA. It is the second abundant class constituting about 10 to 15%. I am telling about IgA. It is secretory antibody and it is present in the sweat, tears, saliva, mucus, colostrum. Colostrum you all know, first milk secreted by the mother after giving birth of the child, and gastrointestinal secretions. Next type is IgD. IgD are mainly found on the surface of B lymphocytes and act as antigen receptors, where it activates B cells for antigen recognition. It is about 0.2% of all antibodies in the blood. Next type is IgE. It is less than 0.1% of all the antibodies in the blood, located on mast cells and basophils, releasing histamine from mast cells and basophils. So these are really involved in the allergic and hypersensitive reactions. Next type is IgG. IgG is the most abundant class of antibodies, which is approximately 80% of the total immunoglobulins. It's found in blood, lymph, intestine. It protects against bacteria and viruses by enhancing phagocytosis and neutralizing toxins, etc. It is only the class of antibody which can cross placenta from mother to fetus, thereby giving immune protection in the newly borns. Next type is IgM. IgM is about 5 to 10% of all antibodies in the blood. It is found in lymph. It's the largest immunoglobulin which is secreted first by the plasma cells. So it is the earliest immunoglobulin to be synthesized by the fetus. So it can't pass through the placenta. So this is all about the different types of antibodies and their functions. In this slide and next two slides, we will study the structure of antibody. So antibody structure, immunoglobulins have been studied extensively and serves as a model of basic structure unit of all immunoglobulins. An antibody molecule consists of following parts. First of all, heavy and light chain. It's Y-shaped structure, and an antibody molecule is made up of 4 peptide chains, 2 small called light chains, and 2 longer called heavy chains. Hence, an antibody is represented as H2L2. The heavy chains have larger number of amino acids, while light chains have smaller number of amino acids. Heavy and light chains may be either lambda or kappa type. This is very important diagram and usually asked in the exams. So second part is constant and variable regions. There are two different regions, the constant region and the variable region in each chain of the antibody. Now next are disulfide bonds or hinge region. A disulfide bond joins a light chain with a heavy chain. Two disulfide bonds also link the two heavy chains. This part of antibody displays considerable flexibility and is called hinge region. Because the antibody arms can move somewhat as the hinge region bends, an antibody can assume as Y-shaped molecule, as I earlier told you. This diagram is showing again the heavy and light chains, constant and variable regions, disulfide bonds and hinge region, and fragment antigen binding and fragment crystallizable fragments. Two identical fragments of Y-shaped molecule possess the antigen binding sites and thus named fragment antigen binding FAB. The antigen binding sites bind to the specific antigen in a lock and key pattern, forming an antigen antibody complex. The third fragment, which lacks the antibody to bind to antigen and can be crystallized, is therefore known as fragment crystallization. So the stem of the Y-shaped antibody monomer is called Fc region. This is the so named because when antibody structure was first being recognized. So this is all about the structure of antibody.